A very good afternoon, Professor. Today I am going to present my personal timetable. Here is my personal timetable that is planned for the next week work. The first one is AO research and paperwork, and also about the literature survey. And simultaneously, I am working in the lab for doing IDC graphene research with Prashant to check the capacitance measurement, and also simultaneously work writing a paper related with this, and also review paper writing on the RF and microwave biosensors, their trends and challenges. And the next Wednesday in the will go for the co-research meeting. This literature survey about the coating of graphene and the graphene oxide on the IDC chip. There are the various techniques and the methods for the immobilization of a graphene oxide on the substrate. There are the various substrate as well also. The substrate is SiO2 and glass substrate, and someone is having using the hard kind of substrate, and also various techniques on the lithography technique and transfer printing technique for the graphene immobilization on their substrate. And also the two things that is considered in the memory technologies, and that is established and the prototypical techniques. And also there's the categorized into two things, volatile and then non-volatile techniques. So it's the various things are to do, and someone is as I've seen that there are various small techniques and there's methods to mobilization method. And first one is just silica substrate and to the graphene oxide, and the someone is using the polymer PVDF for the better attachment on the substrate. And this technique is basically related with the laser induced method. It's a laser induced method is to graphene immobilization through the laser technique, and after that the PDMS coating, and it's basically related with the flexible bioelectronics method. So it's and transition metal dichalconides photodetectors also. There are the various translational research going on with this technique for the photodetectors manufacturing and their applications. Here is my rough idea about my next week lab work. Actually, I have designed one graphene PSA biosensor that is having two different cases, with EDC crosslinker and without EDC crosslinker. With EDC, it means we use the EDC crosslinker in this case 1, and the second case we use the without EDC crosslinker to check the capacitance value in a 5 different concentration solution, to whether is there any changes in the absence of EDC and or not. So once the experiment has been successfully done or executed, then we will proceed to the BATIO3 experiment, because BATIO3 a chip, we already know that we have only two chips. So once this experiment has been successfully done, then I will proceed with that chip, and to check the capacitance value and the measurement. And here is my achievements also. I have flash on the this PPT. And this now current situation is my first paper is submitted in the bioscience and bioelectronics, and waiting for the reviewer comments, and now status is under review. So hope for the best, and waiting for the this month to getting the results and the notification from the editorial committee members. And the third one is the review article that I'm writing in progress. And the second one is the capacitance measured by sensor related with the Alzheimer disease patient samples is already writing in progress. Thank you, sir.